So we are coming back from this break. Online folks, we're going to let the chatter in the room die down a little bit. We're having good conversation, so that's good. All right. So we're resuming this post-break period with a series of discussions on genetics and assisted migration. And Tom Woodham is going to be taking us away to talk about the need for a genetics restoration plan and a dispersed network of field provenance trials to identify stock that is genetically best suited to survive ongoing climate change. So I'll turn it over to you, Tom.
Okay, thank you. Well, let's just start off with this slide. And this is an increasingly common view here in the Southwest. And these photographs are all from Arizona. But you can see that these foundation tree species that really define their respective ecosystems are suffering from stand level die-off. And it's largely because we're in a mega drought that's been ongoing for 24 years. And the local stock is clearly not adapted to what these environments have become. And so restoring with local stock really doesn't make sense. And so that raises the issue of where do you get the stock from to restore such sites. A general pattern here too is that we have the drought kill off a lot of trees, and then we have fire, and then we have flood. And so the sites can become quite altered through this whole process. Next slide.
It was a bad time to get my coffee.
Okay, so the whole process of deciding what stock to use is complicated by the level of local adaptation. And many of our trees with widespread distributions are very locally adapted. And so this is one tree that we've worked on quite a bit, narrowleaf cottonwood. And you can see that based upon our genetic analysis and common garden experiments, we can show that each of these ecotypes is very locally adapted. And what you should see here is that these sites show dark green and pale blue. The pale blue shows what we've determined based upon our genetics-based models are marginal habitat. And we feel it's really a waste of time to even try to plant into these marginal habitats, which are currently occupied by these trees, but in the future will become so unsuitable habitat that efforts to replant them would largely be wasted. Where we should really be putting our efforts in are the climate refugia, shown in dark green. And there we have our best success in restoration. I think this is really important because I'm becoming increasingly aware of large projects that just totally fail, like a recent one in Arizona of a $5 million project, largely failed because there was such low or no survivorship. And then the local adaptation is far greater than what you see here. For example, in Arizona, we have reciprocal common gardens along an elevational gradient. And it turns out that even within an ecotype, there's a lot of local adaptation such that if you, every 1,000-foot increase in elevation is another genetically differentiated population, and you don't want to cross ecotypes, or you don't really want to cross many of these local plantings because you're getting a poor genetic match for what the environment has become or will become in the future. Next slide, please. And this just emphasizes, again, how much genetic variation we see. On the left, we have a number of genotypes all growing in a common garden. And the one on the far left is growing so fast that its growth rate is equivalent to what you would find in a rainforest. And the one on the far right, its growth rate is so slow, it's equivalent to what you would find in a boreal forest. and then you have everything in between. And this is all within one population of trees. And then on the right, we have another common garden in which the trees have been planted as clonal replicates in a block design. And you can see genotype number 1 is doing very well, and genotype number 2 is also doing very well, but it has just a much slower growth rate and a very different phenology. Genotype 4 completely died out, and genotype 3 was pretty poor as well. And it's important to emphasize that the animals, from microbes to vertebrates, really key in on these individual tree genotype differences to acquire very different communities. And this is emphasized in the next slide, where we have about 20 or 30 studies that have looked at individual species and communities of organisms, from microbes to vertebrates. And basically, all of these that are shown are have very strong genetic preferences for individual tree genotypes, and they also differ across populations. The dark lines show different communities that are linked to one another and co-vary, such that if you change the arthropod community, you will get a simultaneous change in the ectomycorrhizal fungal community. And the same thing for the pathogens and the lichens and the soil fungi and the soil decomposing bacteria. They all show very strong genetic relationships with individual tree genotypes and populations. The next slide then kind of looks at a summary of what we know, and I don't have time to go over it, but I would emphasize point number 7 about nurse plants and mycorrhizal mutualists. Mycorrhizal mutualists are also tree genotype dependent. And if you get a good match between the mycorrhizae and the tree, you can bump up your performance and survival by at least a third. And some trees just simply won't survive if you don't get the right mycorrhizal match. And the same thing goes with nurse plants. Some of the trees we work on simply cannot establish without an associated nurse plant. And so very important to consider multiple species plantings. Next slide. And so the one thing we should build, I would argue, is that in addition to having a genetics plan for a restoration site, especially a large one, we should have embedded within that restoration site a field trial. And the field trial just simply uses the best stock that we can find. But then it is grown out, and based upon the field trial, we can improve the genetics model on the surrounding planting areas of thousands of acres. And these also need to be multi-species field trials. Here we have a cottonwood and 2 willows, and how the cottonwood does is very dependent upon its neighbor and the genetics of its neighbor. So this is something I think we really need to emphasize because it's really trying to match the real world, and of which genetics plays a really huge part in what's going to live and what's going to die, especially under a climate change regime. Thank you very much.
Okay. Thank you, Tom. We're going to keep moving forward, but if anybody has any questions for Tom, would you mind saving it until the break and we can ask them then? If Tom, there is one in the room for you, so if you're willing to stick around for a minute before lunch to get that question answered, that'd be fantastic.
Sure.
Great. All right. We are going to transition to Brian Rattini, who is working with the Forest Service, and he's going to talk to us about climate-informed reforestation in Southwestern Oregon. So take it away, Brian.
Hey folks, thanks for having me here. Yeah, so I'm going to be talking mostly about a tool that I developed this last year, and that's quickly becoming the heart of our climate-informed reforestation program up here in Region 6. And that is you can go to the next slide. That is the Seed Law Explorer for Extrazonal Deployment, or SEED. This application is essentially an extension of the Seed Lot Selection tool. It uses the exact same algorithm. with some pretty key differences. But just to walk through the general workflow of how the tool works before I show how we've been using it. Again, it essentially works the same way as a zone-based seed lot selection tool runs. So the first step in A is we extract the range of historic climate variation in a breeding zone or a seed zone elevation band of interest. So this is an elevation band within this seed zone up here in region 6. We then establish a transfer limit based on that range of variation. And this is what we consider our essentially our search radius for assessing matches for present or future climate conditions. And so we just project that range of conditions out in the landscape. That's C here. And that's basically the output of the seed lot selection tool, right? At least for a zone-based run. The key difference here in the seed tool is that instead of doing this for a single seed source, the seed tool actually does this for every potential seed source in the region. So basically, we first looped through and generated seed lot selection tool runs for every single breeding zone, both species specific and generic, and every seed zone elevation band combination in the region under all of the climate projections within the seed lot selection tool. We then use that database essentially as our the results to parse for the actual tool itself. So the tool really is just parsing through those results and assessing match for areas of interest. And so within the tool, you can basically dial up and down how strict you want to be with the area you consider a match. So that's D here. So you can consider only areas that are very close to the mean, in this case, the median climate condition of that zone historically, or you can consider anything within the transfer limit. And then what the tool does is you intersect that with some polygon of interest. And so that might be a project area. It could be the whole forest. It could be a breeding zone. And that flexibility has allowed us to use this tool for a variety of different purposes. The main purpose that we originally developed the tool for, though, was to identify quickly and efficiently the best available climate-informed seed for a given planting site. So for a given area that needs to be reforested. And so in order to do that, we also pulled in the seed inventory for Region 6, with the idea being, if we know where every single seed source can be deployed under present or future conditions, we know where all of our seed lots can be deployed safely under present or future conditions. And so that's directly integrated into the tool. You can go ahead and go to the next slide. I'm just going to go through a couple of ways that we've been using the tool in Region 6 so far. The first of those, as I mentioned, is to identify the best climate-informed seed for a given planting site, so to help fill our reforestation backlog. I work with the Fremont-Wainima National Forest, which is one of the forests that has the largest, well, actually, it is the forest that has the largest reforestation backlog in Region 6 at around 260,000 acres. And they only have enough seed to plant about 10% of that. So they're literally thousands and thousands of pounds of seed short. And so in order to help tackle this issue, we first asked what alternative seed sources would be a good match for these planting targets. In doing so, we can both simultaneously tackle seed shortages and make sure that the seed stocks that we're planting will be adapted to the present day conditions that are actually being experienced in those areas, and hopefully will remain well adapted well into the future. And so this is the actual interface of the seed application. This is what the results look like. On the top here, it's an interactive map that basically just shows where each of these seed sources can be deployed within that project area. So that is the area that's within the transfer limit. So for instance, this blue seed source, that's this elevation band from the Deschutes National Forest. In the bottom left, we can see basically just a summary of the balance of, in this case, ponderosa pine from those seed sources. So from those source zones. So again, this is a snapshot of our seed inventory for those areas that are good matches for this planting site. And so we can quickly and efficiently see how much seed is available and how far that'll go in terms of filling the planting need for this particular project area. And then on the bottom right, this is probably one of the most powerful parts of the tool. Since we've already carried out the projections for each seed source under each of our climate projections. So under present and future conditions, we can essentially look at how well each seed source matches the planting site long term. And so, for instance, this same blue seed source here, you can see it's an excellent match to this planting site under present day conditions, and it continues to be a great match long term through the end of the century. And so this is really the type of trend we're looking for in terms of really well matched seed sources for a given planting site. You can go ahead and go to the next slide. So that's one way we've been using the tool. And I forgot to mention, but that's allowed us to increase the amount of acres that we can plant up to around 25 to 30% of that 260,000 acre backlog. So we're talking like somewhere between 50 to 100,000 acres that normally wouldn't have been planted is now getting planted with climate informed seed. The other way we've been using the tool, at least this last year, was to identify collection areas, high priority collection areas that can help fill that seed shortage when we do get cone crops. And so, again, one of the nice things about having assessed the suitability of all potential collection areas is that we can quickly see what off-forest areas would be a good match for our forest. In this case, for the Fremont-Wainema, most of our really good seed sources are at slightly lower elevation off-forest. And so this helped us establish an agreement with ODF and a bunch of other partners that allowed us to do a really incredible collaborative collection effort this last year, which yielded 2,600 bushels of climate-informed cones for ponderosa pine, and it's looking like based on our extraction results, it'll be roughly an equivalent amount of pounds of seed. So we're talking thousands of pounds of climate-informed seed that will be planted will be helping fill that planting backlog in the coming years. And so this tool has really helped us in these two key ways, but we are looking for ways to expand the tool. One of the ways we're expanding it is outside of Region 6. So I'm currently working with the regional geneticists, Scott Kolpak and Andy Bauer, in Region 1 and 5, so that we can expand this out of Region 6. But really, honestly, I think that we're really just scratching the surface of what we can use this tool for, and I'm really excited to see where it goes. You can go ahead and go to the next slide. One of the things that I've learned through developing this tool, since I'm the one who I developed the tool and I also helped implement it, I think I have maybe a little bit of a unique perspective on how these tools, both the building of it and implementation of it, kind of go together. And one of the things that I didn't know going into it and that I really learned through this process was that these tools really have to be fast, efficient, and simple to use. And the reason for that is, surprisingly, oftentimes our seed sourcing decisions are actually happening on the fly, and we have to be able to carry out these analyses super fast. And so if our tools are not streamlined, they're not going to be used. And so oftentimes it's actually better for a tool to be maybe a little bit more simple and a little less flexible, or at the very least, we should have a suite of tools that allow us to either choose to be fast and efficient or more flexible. You can go ahead and go to the next slide. And the last thing here, I think applying that to the reforestation module in question here, I think that it would be really cool to see all of these various tools that we've all been developing put together in a way that the outputs of one tool can be used as inputs into another tool. And that doesn't exist so far. And I think that's a really unique niche that could be filled in one of these types of comprehensive reforestation modules like this. So I guess that's my 2 cents. But yeah, I'd be happy to take any questions. I don't think I have time, but yeah.
If you wouldn't mind hanging on to it till the next break, that'd be great. Brian, thank you. We're going to push on to the next one. Appreciate your contributions. All right, we're going to move on to Jessica Wright, who's going to talk to us a little bit about whether genetics can inform post-fire reforestation. All yours, Jessica.
Thank you. And thank you so much for the invitation to present today. I would like to congratulate Brian on the great name for his tool. That was a beautiful acronym. In government, I've really come to appreciate acronyms, and that was a really great one. If I get the next slide, please. So we're talking about genetics and assisted migration. And so we've kind of basically heard what this slide says already, but this was from the classic work of Clausen, Keck, and Heisey, who were working in the 1920s on the Stanford campus, and they conducted a big reciprocal transplant, similar to what Thomas was talking about in the previous presentation. And they moved these little yarrow plants from the coast to the Sierra Crest to the east side. And they found that plants really did perform best when they were planted back into the environment where they were found. And this was really one of the first demonstrations that there were differences between populations of plants in nature. And there were a few others slightly before, but certainly California was a very early demonstration that this was a thing in plants, that they are different and they are adapted to the environments where they are from. And because there is a growing in common garden environments, we can conclude that that adaptation is the result of genetics. That is, the gardens serve to remove the environmental variation that that you would see in nature, and in a wild population, you wouldn't be able to tell if two are different because of the genetics or because of the environment. But when you plant them all in the same environment, you can conclude that it's genetics. If I could get the next slide, please. I think Joe already showed this picture from Greg O'Neill, showing a sort of demonstration planting of lodgepole pine in Canada at a low elevation site. And you'll see that the trees on the left are a ton bigger than the trees on the right. And the trees on the left are from the low elevation, similar environment to where they're being grown. And the trees on the far right are from high elevation populations. So the same trend is present in trees as well. And so we have quite a bit of data to show that this is true. But I completely agree with Thomas that more data is always helpful. And if I could get the next slide and talk about our project. Oh, I should have spelled that out. I didn't. This is establishing an experimental network of assisted migration in establishment silviculture. And we've established 35, we're in the process of establishing 35 planting sites. And I'm going to guess this is not terribly different than what Thomas was talking about. These are into areas that are being reforested as the result of some kind of disturbance. Largely that's fire and post-fire reforestation, but it can be other disturbances as well. The project is based entirely on co-production. So at each planting site, we test 4 sources of climates, and that basically testing assisted gene flow. Actually, I want to kind of go on a brief digression here. Since we're in a session on assisted migration, I wanted to say that I think it's really important to be clear about what we're talking about when we say assisted migration. And in my mind, there are 3 levels of it. And what we're talking about here in this project, and I think what everybody's talking about so far is what I would call assisted gene flow, where we're moving populations of a species within the current species range. We're not moving new species into an area, we're just going to populations of that species that may have been locally adapted to a different environment, but not going outside the species range. You can also talk about assisted range expansion, where you're going to move the range just a tiny bit and maybe push things uphill a teeny bit. Or then you could talk about assisted migration, where you're really moving things really far. Like let's plant some Joshua trees in Oregon. And so I think it's really important when talking about assisted migration to be really clear what you're talking about in terms of the level of migration and level of things are moving. So we're talking about assisted gene flow in this test, and we are testing 4 different source climates. And in this site, so we have a current climate, sort of the local seed zone elevation band. We're looking at the early, mid, and late century climates. So there are 4 different climate futures we're projecting out, and basically this test is really, really tightly designed to answer the question: does assisted migration work? Is it a good idea? And then how much farther into the future should we be projecting out when we select our seeds? So we have a little bit more and then quite a bit in terms of climate futures. And so these are all planted into single source plots that are ½ acre. So each of those squares out there are a ½ an acre. And then each ½ acre gets 1 seed source, and that gets replicated 4 times. So a basic design for this is 8 acres. And the plot you're seeing here is a 16-acre plot because we're actually testing 2 different sources of seeds, 2 different establishment silviculture approaches. And these could be things like stock type or spacing. And those are, we work with the local managers in the co-production to ask, well, what questions do you have? What do you want to know? And how can we test that? And so we design that into our design, and it's fully replicated. So each seed source is tested against each of the establishment approaches. And even the basic things like the species is selected by the local land manager. Again, we're in the process of deploying these 35 sites. We're going to be planting 6 sites, I think, this spring. We planted about 6 last spring. That's just in California. And my colleague, Rob Slezak, and others at PNW will be establishing more sites in Oregon and Washington. If I can get the next slide. One of our major goals is to make sure this project has a lot of engagement. We have a website. The link is there. And in terms of disseminating information, we're trying to really engage stakeholders and land managers and the public in understanding this whole concept. So if I can get the next slide. Right, so what we know, I think we actually know a lot, and we can use models to predict how a tree will perform based on the climate where they come from. You've seen a bunch of presentations on this. The cast tool, the seed tool, all use these types of models to predict tree performance. But the more we know about exactly where each seed came from, we can get the best model performance. And agreeing with Thomas, more data is always better. And I do want to state that I think the risk of doing nothing is, or could be, greater than the risk associated with doing something like assisted gene flow. And last slide, I do believe that because the more we know about the precise location of the seed source and the exact climate it's adapted to, I think that if we could work on a tool that helps to manage and maintain seed collection and seed banking records. So this is an example, actually a little screenshot from the CAST tool. This is seed zone 526 at 550 feet. And it shows that there's a huge amount of variation in the actual climates found in that seed zone. That's just the different levels of mean natural precipitation and climate in there. And we need systems to keep precise collection data location throughout the entire collection process and deployment process. And I'm just going to say one more thing, and I'm like spacing out because of time. So basically, and as another speaker said, it needs to be easy and efficient for land managers to use because they're doing a lot of work at the same time, and it needs to be an efficient thing for them to use. And with that, I will end.
Thanks, Jessica. Does anybody have one short one? We've got about a minute before we gotta go for it. Rob, you got microphone?
Thank you for those presentations. That was really fascinating. I haven't thought much about community dynamics and genetics as a barrier to seed transfer. One that I think about a lot as someone who works with the National Park Service is sort of the cultural and political barriers to seed transfer. And we have a number of different organizations represented here, and either now or later in the day, I'd love to learn more about how each of these orgs views the idea of planting seeds from a different location into a environment that's being restored. Because I know within NPS, there's a lot of sort of limitations to the acceptance of that idea. And as we all know in this room, fire is agnostic to management boundaries, and our impact can definitely be amplified by having sort of alignment around approaches. But I wonder if that cultural barrier persists outside of NPS. I'm with Schmidt Data Science for the Environment at Berkeley, by the way, but we're partnered with NPS. Maybe that's not a short question. I apologize.
Okay. Jessica, short answer?
Short answer. Well, that was actually why I wanted to go on that digression about defining what we mean by assisted migration, because I think the first step in being able to communicate well is to communicate well how we're thinking about the problem and what we're really thinking about moving and how novel or not novel the material is that we're thinking about moving. So I think it really is important to use for everybody to be at the same table about the kinds of assisted migration we're talking about. And we are not talking about moving Joshua trees to Oregon. I do have some growing in my backyard in Davis. They were selling seeds at the national park. I couldn't help but buy them. But my point is that I think just being really clear about our language is super important.
Thank you. All right, we're going to keep pushing forward. So